With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello students, the question given is malic acid inhibits the activity of enzyme succinate dehydrogenase. During this inhibition, what happens? Our options are Vmax increases, Km decreases. Vmax remains same, Km decreases, Vmax decreases, Km increases, and Vmax remains the same, Km increases. So firstly, we have to know the terms here. Vmax, what is Vmax? Vmax is defined as the maximum rate of enzyme which catalyzes a reaction. So it is the maximum amount of enzyme that is used to catalyze a reaction, and Km on the other hand, it is the Michaelis-Menten constant. So it defines that how easily an enzyme can catalyze a substrate. So it measures that how easily an enzyme it can catalyze a substrate or work on a substrate. Now here, according to the question, malic acid inhibits the activity of enzyme succinate dehydrogenase. So this is a type of competitive inhibition. In this competitive inhibition, what happens? This malic acid, it sits on the place of this enzyme succinate dehydrogenase, and as it sits on its place, this enzyme succinate dehydrogenase, it cannot bind to the substrate. So as it cannot bind to the substrate, no catalyzing effect is seen. So no catalyzation occurs here because of this competitive inhibition. And here, if you see the graph here, the Vmax in this case, it is not changed because there is still a substrate concentration. So substrate is there, so Vmax is not changed. And malic acid, it inhibits the enzyme. So since the substrate is there, so the Vmax, you can see from the graph that if this was the original curve where no inhibitor was present, now when a competitive inhibitor has been added, you can see that the value of Km, the value of Km has shifted because a competitive inhibitor has been added, but Vmax is there. Vmax, the value is similar. Vmax remains unchanged because substrate is still there. So what we can see that here Vmax, the value of Vmax, it remains unchanged. But the distance, you can see that when a competitive inhibitor is added, at first the blue line, this was the value of the Michaelis-Menten constant, that is Km, but now it is shifted. So here, this Km, it increases. So there is an increase in the Km concentration because it now needs more efficiency to achieve this catalyzing effect. So there is increase in this Km concentration. Thus, amongst our option here, the last option where Vmax remains same and Km increases, this is the right option as we have seen from this graph. I hope this solves your doubt. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.